Okay, I'm Malik Amanov. I'm from Bukhara State Medical Institute, named after Abu Ali bin Sina. Today's lecture is about organ transplantation and rejection. So the earliest attempts of transplantation were performed during the World War II, when pilots have received severe burns during the airplane crashes. It was soon realized that individuals reject tissue grafts from other individuals, and rejection usually resulted from inflammatory reactions that damage the transplanted tissues. And rejection is mediated by the adaptive immune system, because adaptive immune system has specificity and memory, and it is dependent on lymphocytes. So in organ transplantation, the term defines that it's a medical procedure in which organ is removed from one body and placed in another body, right, to replace damaged or irreversibly kind of malfunctioning organ. So the individual who provides the particular organ, or we usually call that organ as a graft, is called the donor, right? The person is giving the organs. That person is named as a donor, and whoever receives that organ, right, that patient is called the recipient. And we have the two types of the graft replacement. I mean, if let's say we are putting the organ in its original position, let's say we are putting the liver on the right upper quadrant where its original location is, and that's called orthotopic transplantation. But if let's say we place the organ somewhere else, let's say we are placing the kidney somewhere in the pelvic cavity, right? That's not the original place of the kidney, and that's called heterotopic transplantation. There are four types of the grafts. Autologous graft, when one individual use different tissues in order to kind of replace missing tissue from the same organism, right? Let's say if I have a skin burns on my legs, on my hands, right? If I place the skin graft from my legs, right? This is example for autologous graft. Now the example could be, for example, coronary artery bypass surgery. For example, either we are using internal mammary artery, right, in order to replace coronary artery from the same individual. But if let's say we are using organs or tissues from the identical twins, right, that's called syngeneic graft. And also we have another type is called allogeneic graft. That's the most popular one, and most of the transplantation surgeries are done on the allogeneic grafts. These are the two genetically different individuals. If let's say I receive an organ from someone else whom I don't know at all, right, that's the example for allogeneic graft. And we also have syngeneic graft. That's the between different species. For example, if a person receives let's say heart valves from the swine, right, that's example of the syngeneic graft. Now, why transplantation is not always successful? Why sometimes we experience rejections? Because it is primarily caused by the strong immune response to non-self MHC molecules. The success of transplantation depends on the donors and recipients' MHC molecules. Those are the major histocompatibility complex molecules. And when donor and recipient differ at the MHC, and alloreactive immune response is directed at the non-self alloantigens, right, those allogeneic MHC molecules on the graft. So with the advances in immunosuppressive medications, immunosuppressive drugs, MHC matching has become largely irrelevant for most allografts. Now, here we have the expression of the MHC or HLA. HLA stands for human leukocyte antigens. They expressed in codominant and polymorphic, right? So because each person has two haplotypes, right? One we receive from a paternal side, another one we receive from the maternal side. And these two sets of the genes, they are located on the chromosome 6. Then expression of these genes is codominant because MHC antigens are expressed on the cell surface in the codominant manner. So as I explained, one from father, one from mother. So also these genes are very diverse, meaning that they are polymorphic. So they have a lot of allelic variants of these MHC class 1 and MHC class 2 genes. Now let's see how we see the induction of the immune response against transplants. Whenever we perform transplantation, right, this is the tissue that has been transplanted to the, okay, this is the kidney, right, that has been transplanted. What happens after the surgery, the antigens of this tissue, this is coming from the donor, right, they are taken, transported by the dendritic cells to the nearest or draining lymph nodes. And you can see here, and this process is called sensitization because these antigens are recognized by the alloreactive T cells which reside inside the lymph nodes. So here you can see the process of activation whenever the dendritic cells, they present alloantigens to stimulate T helper cells, right? Because this alloantigens, right, our T cells has never seen those antigens. They are coming from the donor side, so they become activated and they become alloreactive cytotoxic lymphocytes. These are both are the helper T cells and alloreactive cytotoxic lymphocytes. They are become activated. Now T cells travel to the graft. You can see here they start moving, right? This is the effector and this is the effector, right? And they are going back to the tissue and they are causing the inflammatory reaction, right? Cytokine secretion and the killing of the graft cells. That's the mechanism of the rejection. The first step is the sensitization. Dendritic cells, they are carrying antigens to the nearby lymph nodes. T cells are getting activated and T cells are traveling back to the organ and causing inflammatory reaction. So therefore it's very important to match the donor and the recipient, right? We currently use HLA typing. This is one of the methods that kind of provides the success of the transplantation of the organ, right? So there we have the 5 HLA genes. Those are the A, B, C, D, R, and Q. So one from father and one from the mother side. So overall we have the 10, right? If 10 and 10 matches, all are matched, we call it 10 to 10 match. And also we can do DNA sequencing, and also another method is a cross-matching that's performed cytotoxic and preformed cytotoxic antibodies in the recipient serum against the graft. They are detected by observing the lysis of the donor lymphocytes. Again, pay attention here, those are the preformed cytotoxic antibodies, right? Before the surgery, the patient, the donor, the recipient already have those antibodies. And also we can do ABO blood group compatibility. Here you can see the graft failure rate and the number of HLA matches. Here you can see if we have the 6 HLA match around failure rate is around 30%, right? Sorry, mismatches, right? If we have only one, right, it decreases to 22% of the graft failure. So mismatching are increasing the number, the graft failure also increasing. Now we have the 4 types of the rejection. The first one is hyperacute rejection, the acute rejection, and chronic rejection. So let's see what happens during the hyperacute rejection. It starts immediately within minutes and hours after the blood vessels are connected, right? The surgeon usually detects hyperacute rejection because the grafted organ becomes kind of the pale in color, changed in the color, right? It will be kind of the obvious, right? It occurs within the minutes and hours. This occurs because of the thrombotic occlusion of the graft vasculature, and because the patient already have pre-existing antibodies, right? He or she might be kind of exposed to a different type of the human antigens. For example, the person who has received kind of the blood products from other people, they become kind of sensitized already, right? They usually have the pre-existing antibodies, and those are against MHC and ABO antigens, right? Whenever a person's blood vessels, they are connected, we have already preformed antibodies. They go and attach in the endothelium, kind of to cause inflammation and cause the thrombotic reaction. And, here you can see hyperactive rejection is mediated by the IgM, IgG, and the complement, right? The complement is activated through the classical pathway, and there is activation of C5a, because kind of the attracts neutrophil chemotaxis, and C3b promotes neutrophil attachment, and activation C3a, C4a, C5a. These are all the complement components, right? They activate mast cells, which increases inflammatory responses within the graft. So again, the outcome is the endothelial damage, inflammation, and leading to the thrombosis. So what's the outcome of the hyperacute rejection? It's an irreversible ischemic necrosis, right? It is not a common problem in clinical transplantation because every donor and recipient are matched by blood type and the cross-matched, but may still occur because of IgG antibodies directed against HLA protein alloantigens, because of the blood transfusion. For example, the patient had experienced kind of previous transplantations or the multiple pregnancies, right? All of those might kind of sensitize the recipient, then the recipient might have the preformed antibodies. Now let's see what happens during the acute rejection. So acute rejection occurs within days or weeks after the transplantation and is the principal cause of the early graft failure. It is mediated by T cells and antibodies specific for alloetch in the graft, and injury to the graft parenchyma, and cause the blood vessels as well. Okay, so here you can see the reaction after the acute rejection that usually causes the vasculitis of the graft vessels is the dense interstitial lymphocytic infiltrate. It's mediated by the CD8 cytotoxic lymphocytes that directly destroy graft cells, or CD4 T cells that secrete cytokines and induce inflammation, which destroys the graft, as well as antibodies. So currently we are using the immunosuppressive therapy, which is designed to prevent and also reduce acute rejection. Okay, the 2 types of the acute rejection. First is the acute cellular rejection, which is mediated by helper T cells and CD8 plus cytotoxic lymphocytes specific for graft alloantigens, right, that may cause parenchymal cell and endothelial injury. And the second one is the antibody-mediated acute rejection, that here the alloantibodies bind to HLA molecules on vascular endothelial cells that leads to endothelial injury and intravascular thrombosis. That's very similar to hyperacute rejection. Again, the last one is the chronic rejection. Currently, with improved immunosuppression, the major cause of the failure transplant is the chronic rejection, because now we have very good medications, right? We usually prevent or kind of decrease the rate of the acute rejections. It develops insidiously during months of 2 years, and pathologic changes are very organ specific. For example, the kidney, heart, chronic rejection results vascular occlusion and interstitial fibrosis. In the lung transplants, that shows the thickened small airways, usually obliterates the bronchioles, and in liver transplants, that show fibrotic and the non-functional bile ducts. A dominant lesion is arterial occlusion as a result of the proliferation of the intimal smooth muscle cells, and the graft eventually fail mainly because of the resulting the ischemic damage. So here you can see the graft vasculopathy, right? In the hyperacute rejection, usually it occurs within minutes, right? And here is a usually thrombotic kind of occlusion of the blood vasculature, blood vessels, and very similar process also can occur in the acute rejection, and it takes time to the patient to develop kind of the thrombotic reactions, right? But in the chronic rejection, usually there is a intimal smooth muscle cell proliferation. You can see the diameter of the blood vessel here, right? And just compare it with this one where chronic rejection has occurred, right? It shows kind of very decreased diameter of the blood vessel. Again, it occurs because of the intimal smooth muscle cell proliferation, right? That eventually causes decreased the blood flow and the fibrotic changes in the organs. Now, this is the kind of the table that shows all the types of rejections and the time frame when they occur, the cause and the outcome. And let's see how can we manage the rejection, right? Usually we use different types of the medications to prevent rejections. We call those medications are immunosuppressants that block lymphocyte activation and proliferation and reduce transplant rejection by suppressing the cellular and humoral immunity. The first group of medications, they are called interleukin-2 dependent T-cell proliferation and differentiation blockers. Here you can see this is the antigen presenting cells. This is the plasma membrane of the antigen presenting cells, and this side is a T cell, right? Here you can see antigen presenting cell is presenting the antigen to presenting antigen to T cell receptor. So T cell become activates, right? Produces interleukin-2, and interleukin-2 causes the proliferation of the T cells. It's called the self or auto activation, right? That causes the proliferation. So the point here is either to block interleukin-2 transcription or block the interleukin-2 receptor, right? The first group of medications, they are transcription blockers. So this includes the cyclosporin and tacrolimus. You can see here cyclosporin and tacrolimus. Cyclosporin works by inhibiting calcineurin, right? That whenever we increase calcineurin, right, we decrease the production of the interleukin-2, so that interleukin-2 is produced less and they cannot auto-activate the T cells, right? That might eventually decrease the proliferation. Also, we have the tacrolimus, yeah, that's also a calcineurin inhibitor and also binds FK506 binding protein. The next group is the interleukin-2 receptor blockers. Here you can see this is the receptor of interleukin-2, right? And we usually use sirolimus, or some is also called rapamycin, right? That goes and binds the interleukin-2 activation. For example, that inhibits mTOR, right? Rapamycin works by that way. We also can use monoclonal antibodies such as daclizumab, or basilizumab that directly goes and binds interleukin 2 and blocks its activation, right? So also we have the nucleotide synthesis inhibitors. Those are the purine synthesis inhibitors, right? Mycophenolate, azathioprine, those inhibits the purine synthesis, right? You know that the purines are very important. Adenine and guanine, right? Purines, they are important nucleotides for the proliferation. If there is no proliferation, right, there is no cell activation, there is no proliferation. And also we have the anti-lymphocyte antibodies, and those are the for example OC sorry CD3 specific monoclonal antibodies, and also we use the steroids, right, corticosteroids, to inhibit NFKB and suppress B and T cell functions. Okay, thank you for your attention. Here is the last picture. This is the son who gave his kidney to his father. Then the father is a recipient who received the organ. You can see. Okay, thank you very much for your attention.